I remembered to do that this time. Okay, so I'm recording, and so hopefully everybody can see my slide for Environmental Acoustics Regional Working Group. Can I get a yes from somebody to make sure that we can see the slide effectively on the chat? Okay, great. Thank you. I just wanted to go over 3 slides. Welcome again. This is our second seminar in the Environmental Acoustics Regional Working Group seminar series for the spring. We have one more in April, and we have our big symposium coming up in March. So, let's see. If I go to the next slide, this is going to show our webpage, and what I wanted to bring the webpage up. Let's see if I can do that. You have to go in slideshow mode. There we go. There we go. So here's the webpage, and here's under the activities tab. I want to highlight to everybody. This is where you click to sign up for the symposium. It's free. It has breakfast and lunch provided for the first 100 people. We do have to cap at 100 people because that is what the room can hold. So the first 100 people will get seats. And we go back. And this is really what the symposium structure is shaping up to be. I have not put times on here yet. It starts, I think, at 8 or 8:30. We'll have a plenary speaker start around 9:30 after welcoming remarks. And then we have 4 stakeholder presentations from ONR, Maine Coastal Program, which is the Department of Marine Resources, Forest Energy, Geological Survey. We'll have a lunch, box lunch. We'll have another plenary speaker from UH. We'll have 3 more stakeholder talks, and then a poster session. I encourage everybody to try and bring a poster. Faculty, students, stakeholders, advertise your program, advertise your research, advertise your education program. Whatever you like, as long as it is related to environmental acoustics, it is more than welcome. We will have a special for education program posters. Following on that ASA, I know there was one recently. I know Tom Weber made a poster for UNH, and I know that there were other schools that made posters for their university programs or acoustics offerings. So we can put those in a special section too and have them all together. One other thing I wanted to mention, I didn't put this on a slide, but UNH's and Syracuse jointly is offering CBAS 2018. That application period is open. You just Google UNH CBAS for any marine acoustics graduate students who would like to apply. And that is open until February 28th, accepting applications. You do have to be an enrolled graduate student at either a master's or PhD level to meet the eligibility criteria. It's being held here. Last 2 years ago, it's held every even years. Actually, I'll do this. I'll bring it up so people can see it. You can go right to this website, and there's an apply now button. You can just so this is the website for CBAS 2018. Read about previous CBASs if you'd like. With that, I would like to go to our seminar today. So I'm going to stop sharing my screen. Mike Jech, Dr. Mike Jech from the Northeast Fisheries Science Center at NOAA is going to share his screen. Giving the first presentation. Both of these presentations today are on how to estimate animal density from acoustic data. So Mike will be talking about active acoustic data from echo sounders and sonars, and Danielle Harris from St. Andrews University will be talking about density estimation from passive acoustic data, second half of the seminar. Mike, we see your slide, no problem here. And
Okay.
so, I'd like to introduce Dr. Mike Jech and him take this away. And I'm going to mute myself now. So if everybody else who's not speaking can mute themselves, that would be great.
Thank you, Jennifer, and thank you to the steering committee for the invitation. I'll give a pretty short, brief overview of estimating animal densities from active acoustic data, and we'll see the passive data a little later. I'm down here in Woods Hole at the Northeast Fisheries Science Center, part of NOAA, and my main job is to work with the acoustic surveys that go on down here. So first, just some general slides. At Fisheries, acoustics is a field of applying acoustics, underwater acoustics, to fisheries and management or ecosystem observations. And it is a multidisciplinary field. So we can't do this without a lot of different interactions from a lot of different people. We have the echo sounder. I don't know if people can see my cursor. But the echo sounder transmits and receives electrical energy, sends it to a transducer, which converts the electrical energy to sound. And then that sound is propagated through the water where it interacts with objects. Anything in the water that has a density and sound speed contrast to the water will produce an echo. And then any echoes will get. And the acoustic beam it's a cone shape, and we divide that at different range intervals or range gates to get a sampling volume. And within the sampling volume is what we're looking at to estimate the density of what's in there. We can have a lot of different things in the water. We can have fish, we can have plankton, we can have plants. Even the physical discontinuities are shown to have some scattering effect on the sound. So, but it's a multidisciplinary. So we get, and then that sound goes back to transducer, it's converted back to electrical energy, and then from there, because it's a remote sensing, we have to be able to visualize that. So we take that electrical signal, digitize it, and then we visualize it on a computer. So you got computer visualization, you got electronics and engineering, the acoustics, the physics of the propagation and the interaction of the sound with the animals, and then finally we get the biology and the ecology. That's something that I'm the most interested in, and that's what we want to learn from. As opposed to passive acoustics, with active acoustics, we get to choose the source level. So we can choose the kind of sound that we're actually putting into the water. But that choice also affects what you can see, what you can detect, and what you can learn in the water. So this is just a kind of a general graph from McLennan and Simmons showing that the echo sounder frequency is along the bottom axis, the x-axis, from low to high, left to right. And then the range that you can get from high ranges to low ranges and the sampling volume. So you have an interaction of the frequency that you select for your echo sounder and what you're detecting. So typically, the lower frequencies, you're detecting schools of fish, but also bubbles. So you can detect fish or any animal like a sulfonophore or fish that has a gas bubble will scatter sound at low frequencies. The middle frequencies, typically we're picking up fish, and the higher frequencies are typically used for plankton. There's a lot of overlap between those, but in general, that's what we select frequencies for. And then ranges, if you're picking the low frequencies, those can travel thousands and thousands of miles. But the higher frequencies, you get much less range. So it's almost but the higher frequencies up around the upper hundreds of kilohertz in the megahertz regions, it's almost like light. You're getting your sound, your energy is being attenuated quite a bit. So when you're looking at plankton, often, and you're looking from a shipboard system, you're limited in range. So we aren't able to detect plankton very deep with the shipboard acoustics. So you definitely have some trade-offs when you're picking your frequencies. So how do we estimate the fish abundance and biomass using active acoustics? By far, the vast majority of systems that are used are echo sounders that have a transducer that's looking directly downwards or vertically downwards that are mounted to the keel of the ship. Some cases we can put them on tow bodies. In other cases, they're starting to put these systems on AUVs and gliders and other moorings and other things. But for now, the vast majority of systems that are used for estimates, biomass estimates or abundance estimates are on echo sounders on ships. And we measure this volume backscatter, which is the cumulative backscattered energy from all the individuals within the sampling volumes. If you remember from the first slide, with all the different critters that are in the water, they're scattering sound, and that's the cumulative amount of energy that's coming from all the different animals. One thing, we calibrate our systems, so the systems are calibrated to a standard, which is very important for us for comparing between echo sounders and also for the same echo sounder over time. So if you want to get a time series, which is quite important for fisheries management, you want to be able to calibrate those systems so that you know that any change that's happening in your measurements is not due to your echo sounder or the electronics, it's due to something that's going on in the water. But that also encompasses the survey design, so you need to fully encompass the stock and the population that you're trying to measure, and also catchability. So much like a net, we have acoustic catchability, and that's the detection or the availability of the animals to this echo sound or to the sound. And once we see that, the selectivity. So if you have a flounder that's laying right on the bottom or a skate that's lying right on the bottom, most likely you're not going to be able to detect that animal. So you need animals that are up off the bottom or away from any kind of boundaries, actually. Near the surface is also difficult. And so it makes acoustics very amenable to pelagic animals. We can still detect demersal animals or animals that live near the bottom, but animals that live on the bottom or in the bottom are very hard to detect from standard methods. So we often don't include those. That's what we measure. And then the next thing you need to do is we need to classify that volume backscattered. Because it can be a combination of all different types of scatterers, we need to try to classify or try to proportion the amount of energy to what we're looking at. But once we do that, we can actually use that as a relative index. If we have a calibrated system and your classification schemes are being used over time, that provides a relative index, and that's actually can be quite useful for management purposes. We'd love to take it to the next step, which is actually calculating the numeric density, but that takes a lot more information. Potentially takes a lot more information, and the major piece of information that we need to convert that volume backscatter to numeric densities is the target strength. Now I'll get into target strength a little bit later in the talk. But also you need to know the biology. So you need to know what the lengths and weights distributions are. So if you're looking at adult fish, if you're looking at juvenile fish, if you're looking at larval fish, you need to know a lot about, you need to start to know a lot about the population and the characteristics of the population when you start trying to calculate abundances and densities. But essentially, if you divide that volume backscatter by the target strength, and then you use linear units, you get the numeric density, or units per numbers per unit volume. And once you do that, then you can scale up to regions or areas. Most acoustic surveys are done using a systematic design, which is the lower right image. It's they also call it mowing the lawn. It's much like mapping the seabed. We're just mapping the water column. And this is a case of a survey that was done on the northern flank of Georges Bank in the fall time frame, when the herring were getting ready to spawn. But we can also collect data from ships of opportunity. We've been collecting data on the vessels that have conducted the bottom trawl survey here in the Northeast since 1998. And so on the lower left is a track, a spaghetti track, basically, of the survey that's being done for the bottom trawl. It's a randomly stratified, so all the trawl locations are randomly selected, but the path is not necessarily random. The bubbles are essentially what we've classified as Atlantic herring and in a relative density of those herring. So you can see kind of you get to see a large aerial spatial distribution of what we're calling herring. So the red dots, you see them along the northern flank of Georges Bank, along here, and then also up in the nearshore areas. And I'll talk about these in a little bit, but that's what we've classified as V. So you do some summations or statistics on the area, and that becomes a relative index. Here's an echogram. This is of Atlantic herring on Georges Bank. These herring are in the fall are spawn on Georges Bank, so they queue up in about the month prior to the spawning. And in this case, we'll see this is echogram, 3 echograms, multi-frequency echograms. The top echogram is 12 kilohertz, 38, and then the lower echogram is 120 kilohertz. The seabed is the dark red bottom, or the dark red line, the sea surface, and this big mass of color is a aggregation of herring. Each one of the vertical bars is half a nautical mile, and each one of the horizontal bars or lines is 50 meters thick. So you see this aggregation of herring is longer than 5 miles long, and in some cases 50 to 75 meters thick. So that, it's quite extensive. It's probably would cover maybe the campus of UNH, or at least part of the campus of UNH of just herring. And that's one of the reasons why we picked that time and place, because the herring arcs can be spread out in the summertime, feeding in different areas, but then they come down, or a lot of them come down to Georges Bank to spawn in the fall time frame. Did you also notice that some of the scattering characteristics, the herring among the 3 different frequencies look pretty similar in terms of the intensity? But you notice in the upper right-hand corner of the different echograms, you see a very strong or stronger scattering in 12 kilohertz and less in the 38 and 120. So we start to take advantage of those multi-frequency scattering responses to start to classify what we're looking at. But how do we know what we're looking at? Primarily, we use capture data, trawls or nets. So we trawl with a net, and we capture what's down there, and then we get some of the biology, also lengths and weights and other things, diets, ages, maturity stages, and all that. But that's by far the most common way that we use to tell what we're scattering off of. Optical data is also interesting. Avoidance is a big issue for working in the pelagic realm. We've tried this quite a bit, and you put a camera down and you don't see much, and you turn a light on and you scare the fish away. So in the pelagic areas, the optical data doesn't work as well, but it works really well on reefs, so where you have clear water and you can see things. We also use theoretical acoustic models, so I'll show that in a little bit, where we model the acoustic scattering from the animals. But we also need the biology of what we're looking at. So the seasons, the times of day, things can vertically migrate with night or day, the depth of where they are, inshore, offshore. So different species, some species tend to stay more inshore, other species tend to stay offshore. So knowing a lot about the biology of the different animals that are out there can really start to help you interpret the echograms that we're looking at. And other information, commercial fisheries in our case, we get a lot of information from the commercial fishermen that are out there fishing. They end up spending a lot more time out in the water than we do, and so we can learn a lot from them. Here's an example of a net that we used on the Delaware 2. The old Delaware 2 has been decommissioned. This net's a midwater trawl. It's about 100 meters long. The mouth opening is about 30 meters wide by about 15 meters tall. And you can see the big meshes. It's called a rope trawl. So it's designed to herd fish into the middle, and then capture the fish in the cod end. And actually, I don't know if Pat Sullivan is on at Cornell, but this might be him down in the lower right-hand corner, actually working on the net. I can't remember if it was him or not. But anyway, we also put sensors on the nets, and that's the mouth opening of the net, and the dots, or the pink dots, are the herring. So we can capture, we can place the net right in the school and get a representative sample of what we want, of what the aggregations are. Also, in terms of acoustic models, basically the models, the theoretical models that we've been using are analytical models, and they're essentially backscattering from known shapes. So we have to translate the complex anatomy and morphology of the animals and looking into more simple shapes. So the upper figure is a dissection of an alewife, and we can see the muscle and bone, and then we see a large gonad that's kind of a large pink structure near the stomach. But we also see the swim bladder, which is kind of that silver gray object, long, thin silver gray object. And from those, we can take X-rays And the middle image is the X-ray of not the same fish, but of another alewife. You can see the vertebrae, but you also that swim bladder shows up very nicely. And the swim bladder is a gas-filled organ used for hearing and for buoyancy in fish, and that because it's air-filled, it provides a very strong contrast to the water, so that's where most of the echo comes from. But we have a hard time modeling a very complex structure with the analytical models. You can use numerical models, but I haven't used those much. Other people are starting to use those. They're computationally a bit more complicated to use, but they do potentially give you more realistic information. The models we use in analytical models, they can be used very quickly and provide good predictions of the backscatter. But we convert that fish body basically to contiguous finite cylinders, and we calculate the backscattering from those cylinders. And on the left-hand side is a model, a fish, and then the upper curve, and then the lower curve is a curve from a scattering response from euphausids with frequency. And we notice that where we have the red lines are the frequencies that we typically have on our research vessels, and you see that the curve is relatively flat over the frequencies that we typically use. That's what's in the geometric scattering region, and that response is pretty flat. So we'll see in a little bit if we add more and more frequencies and more points along a flat line, and we're not adding a whole lot of information. So for fish in that geometric scattering region, we're not adding a lot of information. If we look at the krill, those same frequencies were up on the Rayleigh scattering region and up in the transition region, and that is actually informative. So that knowing where that transition region is and the Rayleigh scattering can be quite informative for krill. And for fish, we're also looking at the resonance regions. So if you see that real strong peak up at the lower frequencies, that also can be very interesting, because the resonance scattering becomes dependent on the volume of the swim bladder, and which can be dependent on the size. If the fish are using it for buoyancy, then the volume of that swim bladder can be related to the size of the animal or the mass of the animal. Then we can also look at the scattering model. This is for cod. The frequency is along the axis going along at an angle, but also as an angle of orientation. So we know that as the fish changes orientation with either vertical migration or feeding or whatever else it's doing, that can also change the target strike. And that becomes a strong factor in adding to some of the uncertainty of target strike measurements. The lower frequency we see at the angle of orientation has a minimal effect on the target strength. But as we go up in frequencies, that peak becomes much steeper. So as the fish tilts a bit, that the target strike can change by tens of dBs, which then has an effect on how we translate that acoustic volume backscattering into numeric densities. By using that multiple frequencies, we can start to classify echograms to very simple taxonomic levels. And the upper one is a color-coded classification scheme where the red are primarily krill, and the cyan color is primarily herring that's down near the bottom. Those real patches of kind of orange, yellow, gray, green, we don't know what they are. They may be spawning aggregations of shrimp, but then you see the multiple frequencies down below and how they kind of match up to the color classification. But this color classification now helps us a lot in terms of classifying the volume backscatter and apportioning the echo energy to what we're classifying as krill or to Atlantic herring. And then we can take that, scale it up to a full transect. That's a transect along Georges Bank. One thing we highlight here is we know the Atlantic herring down near the bottom is that cyan color, but the classification scheme that we use is not unique to Atlantic herring. It's primarily picking out or classifying fish with swim bladders. And we know that on the upper water column, up near the surface at about 60 nautical miles, the tick mark at 60 nautical miles, we also have a lot of cyan scattering. So we've had to do a lot of towing, pulling a lot of nets in that area, and we know that they're not Atlantic herring, they're scattering by other animals. We also know that juvenile fish tend to hang out in the upper surface water, so they also have gas-filled swim bladders. And so in terms of our classification, we run it through a classification algorithm, and then we manually highlight what we think are Atlantic herring or other scattering types. And then we can go up from the transect and up to the survey area, to the region. And this is an aerial survey, an aerial representation of the distribution of Atlantic herring, or the echo energy that was apportioned to Atlantic herring. And again, so now we can see in the regional scale where these animals, where the Atlantic herring are located. And again, at this time of the year in the fall, this is from 2000, the Atlantic herring were primarily located along the northern flank of Georges Bank, where they're getting ready to spawn. And up on the other areas in the Gulf of Maine, Jeffrey's Ledge, Platts and Cassius, and tipping these ledges, we saw some herring also. So if we do these surveys over years, we can start to map their spatial distribution and also the densities over time. And again, we can use these data as a relative index, which is what we're doing down here for Atlantic herring. So the next step is to start looking at the target strength, which is the backscatter echo intensity from an individual. And it's hard to measure in situ. And it's the in situ measurements are the gold standard, and they're the best way to translate the acoustic energy to numeric densities, because if you can measure the target strength in situ, that takes into account all the variability and all the different behavior that's going on at that moment. But for schooling fish, it's very hard to do because they're schooling, and you need to isolate the echoes from individuals, which can be quite difficult from schooling fish. From other areas, or you can look at the individuals, but for us, for schooling fish, it's quite difficult. So some of the things we do is we look at ex situ measurements, and then we also look at theoretical models. So this is an example of an ex situ measurement that we did a number of years ago with Ben Reeder and Tim Stanton. We actually tethered herring in a little net, and we spun it around 360 degrees and looked at the backscatter intensity from the individual at different angles of orientation and matched it to the scattering model. And you'll see that at near broadside incidences on all the different panels, we get pretty good agreement between the red line and the black line at the data in the model. So when we start getting at these off broadside incidences is when we start ending up with difficulties in terms of actually matching the model to the data. And that's because you start looking at end effects and other things of the animals. And here's what we call a 3D ambit. This is for a Namibian pilchard. So we can go from 2D, 1D to 2D to 3D. So now we've actually modeled the full, what we're calling the ambit, which is the three-dimensional scattering, backscattering representation from that fish at 38 kilohertz. And which we need because there are other technologies that have a lot of potential. Broadband echo centers, broadband, wideband echo centers is where the echo centers are going. They've been typically narrowband. They greatly increase the amount of information. The spectral content is much wider than the narrowband systems, but we need to find out where those are useful. So again, if we do a lot of work, put a lot of broadband echo centers up in the geometric scattering region for the fish, we're not adding a lot of useful information. We need it in other frequency ranges, especially at the resonance frequencies for fish, but in the transition frequencies for other types of scatterers. We have other swath or other sonars that increases the sampling volume. And for patchy distributions, this can be very useful if we have small schools. But the insonifications angles are not vertical any longer. So we're insonifying the animals at different angles. So we need the target strength at different angles. And that's where some theoretical scattering models come into play that we can use. And also we use some wide area sonars that really increase the sampling volume. And I'll show you some examples of that. They're difficult, they're logistically difficult to deploy, and for quantification. Where the broadband can work is in the resonance, where we have mixed species. So we had herring and say redfish and silver hake and other animals. The biology is different for these different animals, so we may be able to pick out resonance frequencies for those different animals. And those may be useful for separating the acoustic energy from mixes of species. The multi-beam echo sounder, I haven't done much work with this. We have one on the different ships, but that does, see the fan or the swath does increase the sampling volume that we get. So the orange cone would be looking like a vertical echo sounder, and the light blue actually increases the sampling volume. You see, for patchy distributions of animals, it can be quite useful to increase your sampling volume. For very long, extensive layers like we had for those Atlantic herring aggregations in the fall, multi-beam may not be as useful. Then we worked with some wide area sonars. This is an example of one that from NRL, a Navy research lab, using mid-frequency sonar. So 3.75 kilohertz. This is north of Georges Bank, up in some of the areas where we had some more fish schools. This was a paper done by Ben Jones. And the arrows show up potentially fish schools in the area. But you notice now we went from sampling volume of maybe tens of meters. Now we're up around 10, 12 miles in circumference in this case from this. Then we also worked with Nick McCrease and Purnima Rattel down in MIT in Northeastern at the ORA system and the Penn State system that they had. And now we're increasing our sampling range out to tens of kilometers. And this is some Atlantic herring aggregations on the northern flank of Georges Bank from 2006. So we've gone from very small ranges up to very large ranges in terms of measuring the aggregations. I think where we need the most work in terms of classification is, most work in terms of the active acoustics is classification. Fisheries management really wants species level identification. And it's harder to say, well, it's a clupiid versus it's, a skate or something else, or cod or haddock. So you need species level classification. So we need to work on that. Although for ecosystem work, sometimes, classifying to say a fish or a krill is sufficient. But it depends on the needs of people using the data. And again, converting those acoustic energies to numbers, the numeric densities, really working on the target strength information that we have to get more reliable and more consistent density estimates from the acoustics. So I guess we'll wait for questions till the end.
Okay, we have about 1 or 2 minutes for questions. So why don't you, Mike, unshare your screen, and Danielle, you can share yours while, if anybody has a question in this room, or if you want to write in with one question, I can take 1 or 2 now while Danielle gets set up. And then we can do more questions at the end. Thank you so much, Mike. Let's have a round of applause for Mike from our room. I can tell you right now there's about 70 to 80 people online, all amassed in different rooms. We've got about 15 in the room here and a bunch of others from UNH scattered online. So we've got a good audience today. Anybody else have a question? Or we can hold them till the end too. And Danielle, are you okay to share your screen? You have to unmute yourself.
Yeah, I'm just trying.
Okay. There we go. We see your screen.
Okay.
Perfect. So this is Danielle Harris. She's from the University of St. Andrews, and she'll be talking about passive acoustic density estimation. So
Okay.
Danielle, I'll let you take it away.
Thanks, Jen. So just in case St. Andrews is a little unfamiliar, I'm up there in that yellow dot across the ocean, so hello. So for a long time, passive acoustic monitoring has been seen as advantageous in comparison to traditional visual monitoring, simply for some of the reasons illustrated on this slide, that you can take ships out in nice weather, but if it gets gloomy or wavy and stormy, you might be limited. And of course, when it's dark, you can't conduct your visual surveys. And so passive acoustic monitoring has definitely taken off. And the bird there is to denote 2 things. One is sometimes animals are very cryptic, and marine mammals are a very good example of this when you're trying to look at them visually. And also a lot of the things I'll be talking about are suitable for terrestrial species too. So my focus is on marine mammals, but there's no reason why these methods don't work for other species too. So the challenge really is to go from recordings of calls. So I have a spectrogram there of 12 hours of data with a lot of blue whale calls in it. We'll come back to those in a little bit. And we want to take those data, probably run them through an automatic detection algorithm to get numbers of detections. And on the bottom there, you can see 2 years of data at 2 different sites. And then we want to use those detections for maybe many reasons. So density and abundance is just one of them. But we might have behavioral questions, there might be mitigation purposes required, we might have questions about how the animals interact with their habitat, and many others that I'm probably not thinking of. And so we need to think about, though, in order to use our acoustic data, some considerations. So we might want to think about, are any of our detections from our algorithms false, telling us that there's a call there when there actually isn't? We certainly will miss detections, normally those that are far away or quiet. To get density, we need to estimate our surveyed area. And we also really need to, as Mike mentioned, consider the behaviour and the biology of the animal, and in this case, really understanding why these animals vocalise. So just a quick note on why we chase these absolute abundance and density measures. And so it's very tempting to have your number of detections, and here again, here's 2 years of blue whale data from one of the sites I just showed you earlier. And you can see some seasonal peaks in those detections, and it's very tempting to try and make biological conclusions about maybe the numbers of whales making those sounds. But there are several reasons why such a relative measure of abundance might not be telling you the full story. And so what I've done in the next few slides is alter the data artificially to show you a worst case scenario. And so let's take these raw detections. So maybe I've run them through an automatic detection process, and I suspect that sometimes my detections, some of them are wrong. My detector's not perfect, and so I might be able to check some of those and adjust my numbers of detections for those false detections. So in the next slide, you should just see those bars jump a little bit as we've corrected for false detections. So that's done. But then the question is, well, what if I've missed calls? And you could imagine that across these 24 months, noise conditions changed, and so maybe calls were missed, not because they weren't produced, but because they were masked by noise. And again, this is a worst case scenario, but let's say we could correct for the ones we miss, and suddenly our numbers of detections, bringing back in the ones that we've missed, might suddenly look something like this. And so you can tell straight away that your conclusions about the numbers of animals in this area are changing all the time as we correct for these different things. And this was actually demonstrated by work by Tyler Hebley on humpback whales. And I've got some references at the end of this, which you're welcome to have, which showed the importance of considering the missed detections. And then finally, we've got those detections, but what we're really interested in is the number of animals. And so if we knew something about how often animals make the sounds that we've detected, we can convert numbers of detections into numbers of animals. And again, if the animals change the rate at which they call across time, which is perfectly possible, then again, when you convert to number of animals, your seasonal plot suddenly looks a lot different from the plot you started out with. And finally, if you know something about the area that you are monitoring, on the y-axis we had numbers of animals, now we've got animal density. So that's the reason that we go to all of this trouble to convert our detections from passive acoustic monitoring into density, and certainly to think about all those factors that can affect our detections and ultimately our conclusions about the animals themselves. So just a quick note on density and abundance estimators in general. Can I just check? It's gone very quiet. Can you still hear me?
We can hear you, and yes.
Perfect. Okay, good. Just wanted to make sure I wasn't talking to myself.
Yeah, I put everybody on mute but you.
Perfect. Okay. So a little bit about density estimators, just in case you explore the literature, you'll see certainly from the passive acoustic and density and abundance theory in general, these estimators crop up. So the general idea is that to get density, we detect a number of things, so that's little n on our density estimation equation or density estimator. And if we know something about the proportion that we missed, you'll see P with a hat on there because it's something we estimated. and something about if we know the area that we surveyed, which is little a, then we can get to density. So the basic idea goes, you can see in the schematic a pretend ship survey, a systematic survey, just as Mike was talking about for the active acoustics. So imagine that ship, it doesn't cover the whole survey area, but it surveys those blue lines and it detects things, and the black dots denote very realistic animals, and the red dots are those animals that were detected within the survey. And you can see in the green or blue strips that there are black dots there. So there were animals that went undetected. So even on the surveyed area, the blue area, not everything was detected. And so if we know the proportion of those animals that remain undetected, that gives us our probability of being detected, which means we can correct for the animals in all of the blue surveyed strip, as if we saw everything. And then if we know the area that we surveyed, we can work out the density of animals in that area. Furthermore, if we know the total study area, so the whole rectangle, we can go from density in our blue surveyed strips up to abundance across the whole area. So that's a line transect survey, essentially. You may have heard that terminology. So we can look at points instead of lines, and this is something we can consider for acoustics, that you have an instrument listening out over a radius. And so the same idea applies. You could have a number of points that are monitoring, they're in blue, and that you have detections, which are red, but then equally within some listening radius of the instrument, you miss things, and that's perfectly reasonable. And so here's another density estimator, just the same as before, so nothing's changed. But if we're counting calls, not animals, it changes a little bit. So instead of having n being the number of animals we detect, n is now the number of calls that we detect. And because we're listening over time, on the bottom of our estimator, we get a couple more things we need. One is T, the monitoring time, and R is the average call production rate of the animal, or Q rate, because the animal's producing a Q. And that's something that we need to estimate. So if we know how often that animal produces the Q over time, then we can back out the number of the average density of animals in that area. So just a very brief example, and adding in something about false positives just to make it more applicable to passive acoustics. So let's say we have 125 detections in a 1 hour survey. It's really simple. We have checked some of those detections, and we know that 20% of the time our automatic detection algorithm got it wrong. It's telling us that we had a whale call when we didn't. And we also happen to know that the probability of detecting a whale call is 0.4. And we happen to know that this particular type of whale, on average, calls 5 times per hour. So we can use that density estimator to get at the number of whales. So if we have little n being the number of detections, that was 125, we've got 1 minus our false positive rate, which is 0.8, which you can see on the top of the equation there. And all that does is scale down the 125 detections to 100 to say, okay, some of the time we got it wrong, that these weren't actually, these weren't whale calls. And then if we know that the probability of detecting the call, as I said, was 0.4, that scales up those detections, those 100 detections, to say, well, actually, in that hour, we estimate there were 250 calls produced once we think about the ones we missed. If we know the time monitored, T equals 1, and we know the Q rate, 5 in this case, then we can turn those 250 calls into an average of 50 whales making those calls in that 1 hour. So it's very simple, in a sense, to pull all these pieces of the puzzle together and the estimators themselves, but each one can be quite a challenge. So as I mentioned, we need that call production rate. I could talk for a long time about how that's difficult, and there's all sorts of studies going on to better understand the behavior of the animals. As I said, we need to think about, if we're using automatic detection algorithms, that we need to think about false positives and how they perform. False negatives, people, when we talk about acoustics, often you hear about false positives and false negatives. Our false negatives, our missed detections, in general, are taking care of this idea of probability of detecting calls. And the nice thing about these estimators is, for any density or abundance estimation approach, is that any of those parameters that we estimate that have uncertainty or variance, we can incorporate that into the estimator and combine all of the uncertainty of all the different parts of the estimator to give us an overall uncertainty on our eventual density estimate. So we can be very clear about how much we don't know, and we can also pick out which parts of this process we are contributing the most to that variance and can actually guide us on our research questions and where we should focus more effort to improve the estimate of, let's say, the call production rate. So I've very briefly mentioned, oh, there's this thing called probability of detection, and if you know it, you can account for the things that you miss. And so that's what density estimation methods in general aim to estimate, and that's the detection probability, because it's such a key part. And in general, the way you estimate detection probability varies as a suite of methods, and it really depends on how much spatial information you have about your detections. And I've mentioned survey design there, just to say for passive acoustics, that often means how many instruments you're able to deploy and in what configuration. Did you have a localizing array? Did you just have single instruments? How far apart were they? All these things. And as with any suite of methods, there's pros and cons to each. But even if you're not concerned about density or abundance, I think thinking about these methods are useful just to ask questions like, how far out was my hydrophone or microphone monitoring? Just so you can start to think about what area am I drawing my biological conclusions over. And so here I've just got this schematic to demonstrate the type of information that passive acoustics gives us. And so we can have everything from simply detections on a single hydrophone or microphone up to very fine scale 3D localization, depending on your instruments and configuration. And what that means for density estimation methods is that the more information you have, it puts you into sort of what I'm calling the standard methods. And some of you may have heard about distance sampling. Another method's called spatial capture recapture. And they're well-studied traditional methods of estimating detection probability. As you move away from having spatial information down to single hydrophones with detections, you start to have to require what I'm calling non-standard methods, and that's things that have been worked on in the last 10 years or so. And there you require maybe more auxiliary data sets. You might have to do lots of little separate analyses to get everything that you need to estimate detection probability, and generally you rely on more assumptions. So if you can put yourself up using standard methods for this kind of work, then you're in a much better position, but you can do things if you have information that's not as informative about the location of the animal. So like I said, this can apply to many species, and here's just a few examples: birds, frogs, whales, primates. So again, I think I've got the references at the end. And so I just wanted to leave you with an example of why we do find it important to think about these things. And here's a story from one type of monitoring system that Jen knows well. This is in part a project we've been working on together. And this is the Comprehensive Nuclear Test Ban Treaty data set. And these are hydroacoustic stations dotted around the world's oceans whose prime reason for being there is to monitor and detect anybody trying to do cheeky nuclear explosion testing. And so these instruments record 24/7, and very nicely the data are made available to biologists and other people not so focused on nuclear explosions. But it's just an example of an opportunistic data set that exists for passive acoustic monitoring, and there are lots of other examples. So there are many instruments out there, and we're really, part of our motivation for these methods is to get the best out of these instruments, to ask as many questions as we can of the animals. So this is one site of the CTBT network, and in the Northern Indian Ocean, I've got a circle there around one of the stations, and that's Diego Garcia. And I did some work there for my PhD on this blue whale call, nominally known as the Sri Lankan call because it was recorded near Sri Lanka, and I've got 3 examples on this spectrogram. So the part that I was mostly working on and my automatic detector worked on was that high tone, well high, not really that high frequency, but it's 106 or so hertz at the time I was recording them. And the area around Diego Garcia is really variable. So out to the east, you've got the deep Indian Ocean basin that drops to about 5 kilometers. Out to the west, you're heading towards Madagascar and the bathymetry gets much shallower, and you can see you've got also a ridge structure going on. So we were interested in working out how does our detection probability of these whales all around the area, how might our ability to detect them change, thinking about this variable bathymetry. And so you can see on the little plot, hopefully there's a tiny green dot where there's Diego Garcia, and then I've got some yellow dots denoting bearings that I looked at. So I kind of did slices around the hydrophone. And the plot you have there of the nine different graphs are probability of detection as a function of range. And note that each range goes out to 1,000 kilometers. And all I want you to see here is that, number one, those detection probabilities vary depending on what bearing you're looking at away from Diego Garcia, driven by the bathymetry. So for example, if we take 90 degrees out into the Indian Ocean basin, it's basically telling us that any animal out there, our probability of detecting that animal drops very, very quickly. So by 100 kilometers, 50 kilometers even, we'd have no chance of detecting that animal. Contrast that with maybe 270 degrees or 315 degrees, and we can detect animals out to very, very large ranges. So we're dealing with large distances from this CTBTO station, but it's variable spatially. Now I'll take you to another CTBT site, Wake Island in the Pacific. And here we were looking at fin whale 20 Hz pulses, so still low frequency, admittedly lower than the blue whales, but similar acoustic characteristics. But again, we wanted to look at how far away we might be monitoring fin whales. So you might think, okay, similar type of species, similar recording system, typically moored in a similar way. So would we expect the same? Well, this is a bird's eye view now of, rather than taking 8 slices like at Diego Garcia, I've actually got 360 slices here. So the middle is where the instrument is. And what that big grey circle is basically telling us is those grey areas denote where we've estimated detection probability to be 0. And basically we can't monitor at these areas. And so I produced this plot for the fin whales, and I was really surprised because it's looking like I can't hear at all, that detection probability is 0 nearly everywhere. If I zoom in to the actual shape of this monitored area, and again, this is out to 2,000 kilometers, so we were expecting, because it's a CTBT site, to have a large detection capability, but the results of this particular work suggest that that wasn't the case. And in fact, once we estimated the monitoring area for Wake Island for fin whales, and we found that compared to Diego Garcia for the blue whales, we were looking at 2 things. One, very small distances in comparison. You'll see there the colour shows where we can listen for fin whales. The colour actually denotes an estimate of how many calls we would predict to come from that area. But the take-home message is, you can see that the maximum, there's a couple of sites at 150 kilometers to the south of the instrument, but most of them are within 50 kilometers. And further, that area is fragmented. So results are suggesting that between this, very close to the hydrophone and a little bit away, there's actually areas of masking where we wouldn't expect to be able to hear whales at all. And so why this fragmentation? Well, in this case, it's down to the transmission loss. So if we look at the transmission loss, you can see that it fluctuates and oscillates. And so we think that's why we've got this sort of fragmentation near the instrument. But really, it's a take-home message that for a CTBTO system, where you might expect for a similar species the detection ranges and the detection probabilities might be comparable, in this case we saw very, very different results. And that's why it's so important to check when you're working with passive acoustic data, in particular, especially if you're working towards density and abundance estimates. So in conclusion, there's lots of acoustic data out there, both from dedicated surveys and opportunistic data sets. We can get to density and abundance estimates using passive acoustics, using a variety of methods. And if you were planning a survey, we would always say, try and put yourself in one of those standard methods, distance sampling or spatial capture recapture. But if you already have data that are collected and you're trying to do the best with them, then we do have non-standard methods that we could apply. And in fact, the 2 methods both at Diego Garcia and Wake Island were both what we would consider non-standard methods, because we could only get at best bearings to the animals. And a big limitation, but actually a really exciting research challenge, is that we don't actually know much about acoustic behaviour of many marine animals. And so call production rates, to switch from density of calls, which we can get to, and we can think about detection probability of calls, getting call rate to get to abundance of animals is actually a big challenge. So just to finish where Mike started off, passive acoustic abundance and density estimation is definitely also a multidisciplinary field, and so you have to think about everything from your survey design to the behavioural data to how you collect those data, and then finally the analysis that you want to do with them. And that's it. Thank you.
Okay, thank you very much to both of our speakers, and we do have time for questions. So anybody remotely logged in, please type in your questions if you'd like, and I can filter them out one by one. And if there's questions in the room here, you can ask your question or come closer so they can hear you. Do we have any questions? Giving people a minute to write in with their questions here before I let anybody go. I really like the fact that we could complement the passive and the active, so thank you to both. We got clapping coming from UMass. Thank you to both of our speakers. That was a very nice complementary presentation for your seminar. It looks like, and I should say for both of our speakers, with your permission, if you have a problem, please email me. We'd like to make the recording of this seminar able for people to view later. So if that's a problem, just let me know. There is a question from UMass. How do you propagate variances without assuming specific PDFs? I think that probably goes to Danielle.
Okay. So I think, I don't know if I'm getting directly at your question, but so there's different ways that you can estimate variance, and one of the nice ways you can do that without having to assume specific PDFs might be through a non-parametric bootstrap approach. So you can resample your, there's many ways you can resample your data to investigate the variance without having to assume the distributions of specific parameters.
Any other questions? We just have a minute left. We don't have much time left, but we got a thank you from UMass for you. I think we're good. So with that, I'd like to again thank Danielle and Mike very much. And hopefully we'll see a majority of the people who are logged on in person here March 20th at the symposium. And again, right back in this room for UNH people, or again online, April 10th, the second Tuesday of the month, again at noon. Thank you very much.
Thank you.
Thanks, Jen.
Thank you so much, guys. You did great. It all came through crystal clear.
Great, thanks.
Thank you.